Okay, so welcome to today's topic. So last week we looked at gametogenesis. We started looking at embryology. And in embryology, we looked at gametogenesis, which is a process which cells differentiate in order to become germ cells, in order to become sex cells. Okay, so we went ahead and looked at gametogenesis in the female, which is known as oogenesis, and we looked at gametogenesis in the male, which is called spermatogenesis. We also looked at maturation of these gametes and the transport of the sperm from all the way from the testis into the female genital tract, and we went ahead and looked at the transportation a of the oocyte all the way from the ovary to the uterus, both when it is fertilized and when it is not fertilized. Okay, so those were the events of the first week of development. Those were the events of the first week of development. Now let us look at the events of the second week of development. Let us now look at the events of the second week of development. So on day 8, remember we said that after this, after the oocyte has been fertilized, it is going to undergo what is known as cleavage. Cleavage is the initiation of cell division. So cleavage has a few stages. So there's the first stage where there is division, the first division giving us 2 cells, the second division giving us 4 cells, the third division giving us 8 cells, the fourth division giving us 16 cells, the 16 cells that are known as the morula. And we say that this should happen by third day of development, okay? And then we went ahead and looked at what happens now to this morula. We said that this morula, the cells will continue becoming more and more, increasing in number. And as they increase in number, remember there is a zona pellucida around these cells. And so this zona pellucida is hard. So as the cells are increasing in number, there is less space. So the cells start becoming smaller and smaller and become tightly bound together. And that is what we called compaction. Okay. And as this is happening, there is now going to be development of fluids, okay, among the cells. Now, this collection of fluid is going to result in some cells being pushed to one corner of this oocyte, of this fertilized ovum, which is now the zygote. So that fluid, when it is becoming more and more, there will be cells that will be pushed to one corner, and those are the ones that are going to become the future embryo. Okay, so that is the embryoblast. And then we said there are other cells that will surround this embryoblast, and then there will be a collection of fluid in the middle. Okay, so now this whole thing, which has these cells surrounding the fluid. Okay, so we said this is the blastocyst with the cells that will become the future future embryo, which is the embryoblast. And then we have around this embryoblast, we have these cells that will become the future placenta. And this we said is known as the trophoblast. This diagram, when you look at this diagram, we're simply looking at a diagram where one side, this is supposed to be a round thing like this. And there is this part where these cells have been pushed in one portion of this round thing, and then there's this big cavity surrounding it. Okay, but this has been cut so that you're able to see what is happening inside. And so this is the endometrium of the uterus with the uterine glands over there. Okay. So now once this happens, we say that in week 1, all this is happening, such that we have this one, the embryoblast is in that corner. We have that fluid cavity over there, and we have the trophoblast over there. And that whole thing, okay, that whole thing is known as the blastocyst. That whole thing is known as the blastocyst. And that is what we said, that is where week 1 ends, and it ends also with implantation. Okay, we have seen that this blastocyst is implanting right now in the uterine endometrium. Okay, so now on day 8, we are saying that the blastocyst is partially embedded in the endometrial stroma. Like you have seen, they have cut off part of it, and this part is embedded in the endometrium. Okay. The trophoblast has differentiated into 2 layers. So now remember, we said this is the trophoblast. Okay. If you look at this diagram, this is showing this is a better representation of how this blastocyst looks. Okay, it's supposed to be a round thing, so these cells that are around it are known as the trophoblast. They're the future placenta. And then we have these cells that have moved to one portion of this blastocyst, and these are the embryoblasts. They are the ones that are called the embryoblast. Okay, it's the embryoblast, this one. It's going to make the embryo proper, all right? So we are saying that the trophoblast by day 8 has differentiated into the cytotrophoblast and the syncytial trophoblast. What is going to happen is the trophoblast that is around this blastocyst will start differentiating. There will be more cells that will start going deeper into the endometrial stroma. When they go deeper into the endometrial stroma, they're going to lose their cell membranes. They're going to lose their cell membranes. And once they lose their cell membranes, you may look at it like just one whole syncytium with multiple nuclei over there. So these ones that are going deeper into the endometrium, this layer is what is going to be known as the syncytial trophoblast. It's around, all around the blastocyst, okay? And then we have another layer which remains inside, which maintains its properties of having cell membranes around the cells, okay? Now that is what is known as the cytotrophoblast, okay? That is what is known as the cytotrophoblast. So we're saying cells of the inner cell mass or the embryoblast also differentiate into 2 layers. Okay, those ones that were pushed to one cell, to one portion of this blastocyst, will differentiate also into 2 layers. There will be the one that will be nearer to the cavity, okay, is known as this one that is nearer to the cavity of fluid here is the one that is going to be known as the hypoblast. It is made up of small cuboidal cells, okay? And then we have this one that is made up of columnar cells. This one is going to be known as the epiblast. With time, fluid is going to settle in between the cells of the epiblast, okay? Once this fluid settles in the among the cells of the epiblast, this one is what is going to be the amniotic cavity later. Okay, this is what is going to be known as the amniotic cavity later. That is why the cells of the epiblast, these ones in light blue, and the cells of the syncytial trophoblast, this side, are going to be called the amnioblast. Okay, they're going to be called the amnioblast. Okay, so by day 9, we are saying that the blastocyst has embedded deeply into the endometrium. It is now completely in, if you've noticed. This is the endometrial stroma, okay? This is the uterine cavity, and it has completely gone in. And at the point where it had entered into the endometrial stroma, we just have this plug that is known as the fibrin coagulin. Okay, this is what happens on day 9. The trophoblast at this at the embryonic pore, this is the embryonic pore. So the trophoblast at this embryonic pore will develop these vacuoles, these cavities. Okay, there'll be all these cavities, which are going to be known as lacunae. Okay, these are going to be known as lacunae. Now, these lacunae will eventually start coming in contact with the blood vessels of the maternal blood vessels, and they will be filled with blood. Okay, they'll be filled with the blood. But before they are filled with blood, they are known as the lacunae, and this lacunae, and this stage is therefore known as the lacunae stage. Okay, on the abembryonic pore, on the pore where the embryo is, where there is no, the embryo is not, which is the abembryonic pore, there will be flattened cells that are going to form. Okay, these are thought to be coming from the hypoblast here. So these flattened cells are going to form, and they're going to line the cytotrophoblast. They're going to line the cytotrophoblast inside. Okay, now this membrane together with the hypoblast will form what is known as they will form what is known as the exocoelemic membrane. The exocoelemic membrane. So this thing in yellow is going to be known as the exocoelemic membrane. The exocoelemic membrane lines the exocoelemic cavity, and because we still have this cavity here. So we're saying this is what is going to be known as the exocoelomic cavity, and it is going to be lined by the exocoelomic membrane. This exocoelomic cavity is what is going to later become the primitive yolk sac. Okay, that is what is later going to become the primitive yolk sac. Now, by day 11 and 12, the blastocyst is completely embedded in the endometrial stroma, and the surface epithelium almost entirely covers the original defect, meaning at this point, it has embedded completely, okay, into the uterus, and this part is almost completely covered, okay. Now we are saying that there is going to be a small protrusion in the uterine wall, okay, which you can see in the uterine cavity. The trophoblast is characterized by lacunae. Remember, we talked about lacunae already. So the trophoblast is characterized by lacunar spaces in the syncytium, okay, in this dark green portion where the cells have merged, all right? And these form an intercommunicating network. With time, they're going to form an intercommunicating network, and it's most evident at the embryonic pore where the embryo is. So the cells of the syncytial trophoblast penetrate deeper into the stroma, okay, and erodes the endometrial lining. Remember, we said there are blood vessels in the endometrial lining, okay, and the syncytial lacunae become continuous with the sinusoids. These blood vessels in the endometrial lining are known as sinusoids, okay? So the lacuna will become continuous with the sinusoids, and the maternal blood will enter the lacunar system. As the trophoblast continues to erode more and more sinusoids, maternal blood begins to flow in, okay? the maternal blood will begin to flow in and through the trophoblastic system. This is what is going to form the uteroplacental circulation. Okay, this is what is going to later form the uteroplacental circulation. Now, there's a new population of cells which appears between the inner surface of the cytotrophoblast and the outer surface of the exocoelomic cavity. Okay, so remember, we talked about the exocoelomic cavity here being the primitive yolk sac, and then we have talked about the cytotrophoblast here. Now, between this cavity and the cytotrophoblast, we are going to notice that there is going to be developing a new layer of cells. Okay, a new layer of cells is going to start growing. So these form a fine, loose connective tissue, and this is going to be called the extraembryonic mesoderm. This is what is going to be known as the which is going to be known as the extraembryonic mesoderm. Now, this extraembryonic mesoderm eventually fills all of the space between the trophoblast externally and the amnion and exocoelomic membrane internally. Remember, this side we have the amnion, okay? And then here, this side we have the exocoelomic cavity. So external to these two cavities, this space will be completely filled with the extraembryonic mesoderm. With time, large cavities will develop in this extraembryonic mesoderm, and these become confluent, meaning they start becoming one big cavity, and they form a new space. And this new space is going to be known as the extraembryonic cavity. So from extraembryonic mesoderm to extraembryonic cavity. This cavity is also known as a chorionic cavity. Okay. The extraembryonic, this space surrounds the primitive yolk sac. We said this is the primitive yolk sac, the exocoelomic cavity, and the amnion, everywhere except at the point where there is going to be communication, except where the germ disc, this germ disc, is connected to the trophoblast by a connecting stalk. That connecting stalk is later what is going to become the umbilical cord. The extraembryonic mesoderm lining the cytotrophoblast and amnion is called the extraembryonic somatic mesoderm. The extraembryonic mesoderm lining the cytotrophoblast and the amnion. So if you look at this side, we said this is the amnion, this is the cytotrophoblast. So this extraembryonic mesoderm on this side, this is what is going to be known as the extraembryonic somatic mesoderm. The one that is lining the cytotrophoblast and the exocoelomic cavity, or the primitive yolk sac, is going to be known as the extraembryonic somatic mesoderm. The extraembryonic splanchnic or splanchnopluric mesoderm, okay? Extraembryonic splanchnic mesoderm or extraembryonic splanchnopluric mesoderm. So the cells of the endometrium undergo decidual reaction. Decidual reaction means they become polyhedral and they are loaded with glycogen and lipids, okay? And this tissue starts becoming edematous, okay? Now, by day 13, the surface defect in the endometrium has healed. Occasionally, some people may develop some bleeding on that particular day, okay? So at the implantation site, this pregnant woman may develop a little bit of bleeding, okay? Now, this is because day 13, post-fertilization, is almost the day that this person, now because that day is almost the day that this person is supposed to have their menstrual period. So you'll find that this woman may confuse the days and think she has had her period. And when she comes to your clinic and she tells you, no, my last period was on such and such a date, but that could have been implantation bleeding. So some women may get confused because of this implantation bleeding. The trophoblast is characterized by villus structures. So at this point, we are talking about this trophoblast being characterized with or by villus structures. The cells of the cytotrophoblast will proliferate locally, okay, and penetrate into the syncytial trophoblast. If you see what is happening here, you're going to notice that these cells of the cytotrophoblast will start entering the syncytial trophoblast, okay? This forms cellular columns surrounded by syncytium. Okay, so you find that they're going in like that, going deeper and deeper, but around them you'll find this deep green layer, which is the syncytial trophoblast. So they are still, the syncytial trophoblast on day 13 is surrounding the cytotrophoblast as it penetrates, as it penetrates the as it penetrates these cells, the cells of the cytotrophoblast will start invading the syncytial trophoblast and they'll start forming columns inside. These columns will still be lined with the syncytial trophoblast. Now these will be called primary villi. So primary villi are the ones that will only have cytotrophoblastic cells and syncytial trophoblastic cells on the outside. That is what happens on day 13. Meanwhile, the hypoblast, remember those two layers of the embryoblast, we have the hypoblast and the epiblast. So the hypoblast produces additional cells that migrate along the inside of the primitive yolk sac. Okay, they migrate from the hypoblast again. We have cells that will migrate. And then when they do that, they will gradually form a new cavity inside this new cavity, and this new cavity is going to be known as the secondary yolk sac. Okay, this new cavity is going to be known as the secondary yolk sac or the definitive yolk sac. This yolk sac is much smaller than the original primitive yolk sac. Okay, much smaller. During the formation of the secondary yolk sac, there'll be large portions of the exocoelomic cavity that will be pinched off. Okay, and these will become exocoelomic cysts. One of these cysts is this one. Okay, these are normally found in the extraembryonic xylem or the chorionic cavity. Okay, we are going to find them. Meanwhile, the extraembryonic xylem expands. Okay, the extraembryonic, this extraembryonic cavity that we had earlier talked about, it expands and forms a large cavity. And this large cavity is what is going to be known as the chorionic cavity. The extraembryonic mesoderm lining the inside, okay, the extraembryonic mesoderm lining the inside of the cytotrophoblast is known as the chorionic plate. So if you look at this, we have, this is the extraembryonic mesoderm that formed, okay? And then now, because there is this huge cavity here, we have this one, okay? This layer of the extraembryonic mesoderm, and then we have this layer of the extraembryonic mesoderm. So we're saying that this layer, which is lining the cytotrophoblast, is known as the chorionic plate. This is the chorionic plate. The only place where the extraembryonic mesoderm traverses the chorionic cavity is in the connecting stalk. okay? In the connecting stalk, that is the only place where you're going to find that the cavity is traversed by the extraembryonic mesoderm, okay? And we said that this connecting stalk is what will later become the umbilical cord, okay? So that is our day 13, and so day 13 and the second week ends. Now we look at the third week, okay? The third week of development. In the third week of development, we have the main thing that happens there is what is known as gastrulation. Gastrulation is where there is establishment of the three germ layers. These three germ layers include the ectoderm, the mesoderm, and the endoderm. Okay, so you find that in the third week, there is establishment of the three germ layers. Now, this gastrulation begins with formation of the primitive streak. Now, remember, we said if you look at this in the second week, this is where the layers are only two. We have the epiblast and the hypoblast, okay? And so this layer, if you get it, and now you're looking at this embryo from the top, you put it here. You remove this, the amnion, you remove it, this layer. Remember, this is the amniotic cavity, and this is supposed to be the definitive yolk sac. So if you remove this membrane that is completing the amniotic cavity, and you remove this membrane which is completing the yolk sac, and you get this double layer here, and you view it from the top, this is what you're going to see. So this is supposed to be continuous here to make a cavity on top, and this is supposed to be continuous to make a cavity at the bottom. So now what is going to happen in week 3 is that there is going to be appearance of what is going to be known as a primitive streak. This is going to be on the surface of the epiblast, on top, on the epiblastic layer, on that layer that is closer to the amniotic cavity. In a 15 to 16-day embryo, the streak is clearly visible as a narrow groove with slightly bulging regions on either side, okay? So you are going to see that it is going to be a narrow streak. There's going to be bulging regions on either side. The cephalic end of the streak, okay, on this side, there is going to be what you're going to call a primitive node on the cephalic end towards where the head is going to be. There's going to be a node. There's going to be a node, and this node is going to consist of slightly elevated area. If you see this, okay, we are looking at this streak towards this end. So that is the primitive node. This primitive node is going to be made up of slightly elevated area and a small pit in the middle, which is the primitive pit. The cells of the epiblast will start migrating towards the primitive streak here. So the cells will start moving and coming into the primitive streak inside, and they're going to become flask-shaped. They're going to detach from the epiblast, and then they're going to slip beneath the epiblast, okay? Now, this inward movement is known as the invagination. This inward movement of the cells from the epiblast going beneath it is known as invagination. Once the cells have invaginated, some will displace the hypoblast, creating the embryonic endoderm, okay? So you will notice that they will come and displace this hypoblast here, and they will form the endoderm. Others will lie between the newly formed endoderm and the epiblast, okay? And so these will be called the mesoderm, okay? And then we have cells remaining in the epiblast over here. Will now change the name from the epiblast and will now be called the ectoderm. Therefore, it is safe to say that all the 3 germ layers come from the epiblast, okay? All the 3 germ layers come from the epiblast. So we're saying as more and more cells move between the epiblast and the hypoblast layers, they begin to spread laterally. Because remember, this diagram is showing us now where we have cut this embryo right here, and we're able to see what is happening there. Okay, so now these cells will start migrating now laterally, okay, on the lateral side. Others will start spreading laterally, others will start spreading cranially, okay? Gradually, they migrate beyond the margin of the disc. They migrate beyond the margin of the disc and establish contact with remember the extraembryonic mesoderm, okay? The one that will be covering the yolk sac. Okay, they will establish contact. If you look at this diagram, we have this extraembryonic mesoderm. Okay, so some of them will establish contact with the extraembryonic mesoderm, which is covering the yolk sac. Okay, so in the cephalic direction, they pass on each side of the precaudal plate. And the precaudal plate is a plate that those are just the first cells. So it's derived from the first cells that migrate into through the node, the primitive node in the midline and move in the cephalic direction. Okay, it is just the first cells that will migrate through the primitive node. They move in the cephalic direction and they form a small plate, which we'll be able to see. So this precaudal plate is going to be found between the tip of the notochord. We're going to look at the notochord as well. This black thing is the notochord. Its function is just to allow the body to know the midline. So it's the one that determines the midline as other organs and systems develop. This is the notochord, which is going to help in determination of the midline. So the precaudal plate is supposed to be somewhere here, okay? So it is found that between the tip of the notochord and the oropharyngeal membrane. The oropharyngeal membrane is just a place where the ectoderm and the endoderm, they are tightly bound together. You don't find the mesoderm here. And this is what is going to be the future oral cavity, okay? The future oral cavity. So now, formation of the notochord. Remember we said that the notochord is one structure that allows for knowing the midline of this embryo, for allowing the midline of this embryo. So prenotochordal cells invaginating in the primitive node. So if you look at this diagram, now we have cut we have cut this developing embryo in the midline, and we want to see what is happening. Okay, so this is supposed to be the cephalic end, and this is supposed to be the caudal end of this embryo. Okay, so the prenotochordal cord cells invaginating in the primitive node move forward cranially. So they will migrate here and then they'll start moving forward cranially in the midline until they reach the precaudal plate, this precaudal plate here. They become intercalated in the hypoblast, okay? So that for a short time, the midline of the embryo consists of 2 layers that form the notochordal plate. Okay, so this is the notochordal plate. As the hypoblast is replaced by endoderm cells, remember the hypoblast here, moving at the streak, cells of the notochordal plate proliferate and detach from the endoderm. Okay, these cells will some of these cells will proliferate and detach from from the endoderm. Okay, as the hypoblast is displaced, okay, as the hypoblast is replaced by endoderm cells, moving in the straight cells of the notochordal plate proliferate and detach from the endoderm. Okay, so these ones, remember, some of these cells in the middle, they're going to proliferate, and they're going to be called they're going to be, initially it's going to be called a notochordal plate. However, when they detach completely, they detach completely and form this. This diagram is showing us a situation where we have cut this disc at this level. Okay, we have cut this disc at this level, okay, so that we are viewing this disc from this end, and we're able to see what is happening on this side. And so we are saying that these cells initially will form this plate, the notochordal plate. With time, they're going to detach, and they're going to form a tube-like thing. So it's going deeper that side and coming on this side, because we are viewing this side, this disc from the cephalic side. Okay, so this tube is now going to be known as the notochord. Then the notochord, this is going to underlie the future neurotube. The neurotube is supposed to be right in the middle, where your spine is going to be, the vertebral column, where the vertebral column is going to be. So now, meanwhile, the development of the trophoblast. So by the beginning of the 3rd week, the trophoblast is characterized by primary villi. We've already talked about the primary villi being a core of cytotrophoblastic cells surrounded by syncytial trophoblastic cells. Okay. Now, mesodermal cells, remember we have this now, the extraembryonic mesoderm, it starts following. And so these mesodermal cells, they penetrate the core of the primary villi, they penetrate inside. And once that happens, they now become the secondary villi. So this is the formation of the villi. Okay, so we have, first of all, the primary villi with only cytotrophoblastic cells surrounded by syncytial trophoblastic cells. Within the 3rd week, we're going to see a situation where the mesoderm is going to start penetrating the cytotrophoblastic cells, and then this is going to be known as the secondary villi. And then with time, the mesoderm usually is the one that contains cells that are stem cells for blood vessels and blood. So with time, you're going to find that the mesoderm is going to differentiate, and there's going to be appearance of blood vessels inside the villi, the secondary villi. And when that happens, that will now be called the tertiary villus. So by the end of the 3rd week, mesodermal cells in the core of the villus begin to differentiate into blood cells and small blood vessels. This is what is going to form the villus capillary system. The villus is now known as a tertiary villus or a definitive placental villus. Capillaries in tertiary villi make contact with capillaries developing in the mesoderm of the chorionic plate. Okay? Now, capillaries in the tertiary villi will make contact with the capillaries developing in the mesoderm of the chorionic plate. Remember the chorionic plate being this extraembryonic mesoderm that lines the syncytial trophoblast. Okay, so there's capillaries that will be there developing in the tertiary villi. They'll start making contact with the capillaries that will form in the extraembryonic mesoderm. as well as in the connecting stalk. So as well as in the what? In the connecting stalk. These vessels established contact with the intra-embryonic circulatory system with time, and this will end up connecting the placenta to the what? To the embryo. So we find that there will be now a connection between the blood circulating in the placenta with the embryonic blood over there. Okay. So when the heart begins to beat in the 4th week of development, the villus system would have already been ready. Okay. It is ready to supply the embryo proper with essential nutrients and oxygen. Meanwhile, the cytotrophoblastic cells in the villi, remember we are saying that these villi now, so now this villi will keep developing. The cytotrophoblastic cells will continue invading the syncytial trophoblast all the way until they go and reach the maternal endoderm. Okay, they will continue going there. And once they reach the maternal endoderm, they're going to be spreading so that the cells, the cytotrophoblastic cells will go on this direction. And these cells are going to meet with cytotrophoblastic cells from the other villi, completing the whole circle. And this is what is going to be known as the cytotrophoblastic shell. Okay, the cytotrophoblastic shell. This is the connecting stalk, the future umbilical cord. This is the chorionic cavity. Okay, this is going to be our future placenta. All right. This shell gradually surrounds the trophoblast entirely. If you look at this now, we have the syncytial trophoblastic cells surrounding the trophoblast completely, and that attaches the chorionic sac firmly to the maternal endometrial tissue. Okay, villi that extend from the chorionic plate, okay, from this chorionic plate, and to the decidual basalis, which is the decidual plate, villi that extend from the chorionic plate to the decidual basalis, that is the decidual plate. The part of the endometrium where the placenta will form are called the stem villi. They are called the what? They are called. So the villi that extend from the chorionic plate to the decidual basalis. There are some villi that will extend all the way from the chorionic plate to the decidual basalis. Those are known as anchoring villi or stem villi. However, you find that there will be others that will branch from the sides of the stem villi. For example, you look at this villus, it's going to have some branches. It's not going to reach all the way to the maternal side, to the cerebral basalis, and so those are known as free villi. Okay, those are known as free villi. So the chorionic cavity, meanwhile, becomes larger, and by the 19th or 20th day, the embryo is attached to its trophoblastic shell by a narrow stroke. Okay, the connecting stalk later develops into the umbilical, into the umbilical cord. And this one is the one that forms the connection between the placenta and the embryo. Okay, so that is what we are saying. So this will be happening by 19th and 20th day. So in the next session, we're going to look at what happens now after the 20th day. For now, we'll end here.